Okay, looks like we're live. Thanks for joining me, Darko. I think we should give 1 to 2 more minutes before we have people join us, or before we actually get started, and that should give enough time for people to join us. So, if you just entered the room, thank you for joining us. Darko and I will begin our discussion around Estimands in probably 2 minutes' time. So sit tight for the next 2 minutes. Thank you. Darko, while we wait for people to enter the room, how's your day going?
Day is going really well, and thank you so much. And it's really great to join you on this very important discussion. Fantastic topic, and while we wait, I think that the audience that is already in, we can just provide some information that we will be answering questions live after sometime before the end of the webinar, and the rest we will also answer after the webinar.
Yes, that's a great point. So we're going to get started here in about a minute. We're just giving a couple more minutes for people to enter the room. And then throughout today's discussion, we will take questions. We'll hold our answers until the end. But please, as we discuss, if you come up with a question, enter it into the chat. We'll give 20 minutes, roughly 20 minutes for Q&A at the end of today's discussion.
While we wait, how was your day, Robert?
It's good. It's good. So I have a dog. He's very fluffy. He's a Samoyed. He's very fluffy and white. And it decided to downpour on us today here in Chicago. So halfway through the walk, and I did not check the weather, and nor did I look up. And we just both got soaked very early this morning. So I'm dealing with a very wet dog who. No amount of towels on earth will get him dry. So just trying to keep him off the couch, but he's a good boy. So it's a good day. It's a good day. Awesome. Okay. I think we've given enough time to have people enter the room. So thank you, everybody, for joining Darko and I as we discuss for the next hour a very important topic that's becoming more and more relevant in clinical trials in the life sciences, and that is Estimands. So hopefully today, what you'll take away from our discussion is, what are Estimands? Why do we need them? How do you set them up? How do you report them? And in order to facilitate that goal, Darko and I have a lot of discussion points that we'll go through, and we also have some examples of real studies that have used Estimands, and we can show you exactly what they look like, how they were calculated, how they were designed, how they were calculated, so on and so forth. So that's what we're doing for the next hour. Hope you're comfortable. Please grab a snack as we dive into this topic. But before we do, maybe just some very quick introductions. Darko, would you like to introduce yourself to the audience?
Of course. So I'm a clinical biostatistician. That is my main profession. But I also work in different fields such as machine learning, artificial intelligence, and so on. But as I said, today we will be focusing on a clinical research topic, on clinical biostatistics topic, and I think it will be a very interesting topic. But before we really start with the Estimands, would you like to say a few words about yourself, Robert?
Sure. Thanks, Darko. I appreciate that. So Robert Ratchford, I am a biostatistician by training. I have a master's degree in biostatistics, and I've been in the life sciences industry for the past 15 years. I currently serve as the Vice President of Biostatistics and Statistical Programming at ProPharma. We are what I would say a mid-size RCO, so a research consulting organization, and we are very wide in the services we provide. From a statistics standpoint. So we can help design studies, we can help execute studies, we can help report studies to the FDA. And one of the topics that comes up in my everyday life very commonly is Estimands. And so earlier this year, January of 2024, I started a side project called Better Biostatistics. It is meant to provide free resources to anybody and everybody who wants to learn more about statistics and programming. So it's focused on statisticians and programmers, but it's also providing material to medical affairs professionals, executives who need to understand what their biostatisticians are doing, or maybe clinical operations personnel who want to know more about how they can interact better with biostatisticians. So today's webinar is a great step in Better Biostatistics' journey towards helping to educate the life sciences industry as a whole as to what statisticians can do, specifically when it comes to Estimands. A little bit more about me. I live in Chicago, and I currently have two daughters. One was just born, so I'm so fortunate. And we have a dog, like I was saying earlier. So that's a little bit about me. Let's get this rig rolling. Darko, would love to start with you. Talk to us about Estimands. Give us the high-level overview of what these things are, why we need them. Take it away.
Fantastic. Thank you so much, Robert. And as I said, I think this will be a very interesting discussion, very useful for the audience, because it will be more casual. We will also show some real-world examples. We will discuss why are Estimands such a trendy topic, but also a topic which is important for the whole clinical research and pharmaceutical industry. So let us start with the basic question: what are estimands, and why are they important? So every result in clinical research has something that I would call a construct that is related to that result. So if we observe a result as a hazard ratio or a mean difference, it doesn't matter. Every result has a whole world that's behind that result. So it's not just a number. There is a whole world of clinical, statistical, biological aspects that lead to that result. And estimands are exactly about relating what we are estimating, so the quantity that we are estimating, the result that we are estimating, with that construct, with all those clinical features, with all those biological and statistical features, wrapping them up into one systematic definition so that we can accurately describe the result and accurately describe the estimate that we are making. So from a definition perspective, that would be something that I think is important for the audience, but it's also important for us biostatisticians, right? Because estimates are defined in some of the most important documents by the FDA, EMA, international harmonization documents, and so on. So they are very important from the regulatory perspective as well, from the perspective of standardizing and harmonizing our research. And finally, estimates are very important to avoid confusion with interpreting the results. They're very important and helpful for all researchers to be more accurate when they define what is the treatment effect, what is the result, how do we interpret in relation to different clinical and biological features. So not just statistical ones, but also clinical and biological ones.
Would you mind if I shared a very quick slide that I have that I think exemplifies exactly what you're saying, Darko? So
Please.
yeah, hopefully the live audience can see this, but everything Darko just said is perfect. That's exactly what an estimand is. And really, when you conduct a clinical trial, you want to answer a very specific question. Does my drug do this better than what's currently on the market? Or will this drug be safer than what's currently on the market? That's kind of the general high-level type of question. And you ask that, and then you conduct this clinical trial, and you think that the clinical trial is this perfect utopia of data, right? Where it's all clean data, nothing bad happens to the subjects, everything is good. And so in this first case up here, really what you're seeing is the utopia or El Dorado version of a clinical trial, which never happens, right? We are humans. This is the real world. Things happen. And so the question you ask oftentimes gets muddled. It gets altered by these things that we call intercurrent events, or ICEs. And that's a very key term to know when dealing with estimands. So an estimand, really what it is it's saying, okay, if I ask a clinical trial question and the study happens perfectly, great, no problem. I can interpret those results exactly as Darko said. It's easy to interpret those results. But if an ICE occurs, which they almost always do, things always happen, then all of a sudden my interpretation of those results becomes very different. And maybe we don't know that. Maybe it's so subtle that we have lost sight of really what's going on in terms of the clinical trial question. And so what we've developed, or what the International Council of Harmonization has developed through their ICH E9R1 guidance, which is the key document that we'll be referencing throughout today's presentation, what they've identified are these things called estimands, which are basically the ability to plan for an ICE that's going, you know that it's likely to occur. The subject will withdraw from the study, for example, for whatever reason, or the subject will take, the patient will take other medication that kind of isn't necessarily in line with the protocol. Those things are likely to happen. And so the estimand allows you to plan accordingly to handle when those situations actually occur and make your results easier to interpret. So a couple of key things to note as we continue on with this discussion. ICEs, if ever you hear us use the abbreviation ICE, we're talking about an intercurrent event. That's just the thing that happened during the trial to that subject that makes interpreting the results a little bit more difficult. And then the estimand, all we're really using it for, its main purpose is to make our results much easier to interpret. Darko, what do you have to add to that, please?
Well, I think that's a really fantastic explanation for the audience. And you pointed out one very important segment, which is the intercurrent events. So as we will discuss later, we will of course explain other segments of the estimand. But intercurrent events are probably, if we had to choose, I mean, they're all important, but if we had to choose, intercurrent events are really the most important segment to think about when dealing with estimands. Because most other segments, anyone who is in the research field has been dealing with other segments, such as population, variables, and so on. But intercurrent events are something that's not easy to understand. So obviously we will try to facilitate understanding of this for the audience. So the intercurrent events, as Robert explained, they happen most of the time, especially in larger studies, right, where there are many patients. The probability of these intercurrent events, non-adherence to therapy, other types of intercurrent events which may cause the results to change in relation to how we view these intercurrent events. So if you, as example, if we view an intercurrent event using approach A, we may have one hazard ratio. And if we use a different approach B, in relation to intercurrent events, that hazard ratio may change. It may be a whole different result, depending on which angle do we take in relation to these intercurrent events. And obviously, researchers have developed different strategies to deal with these intercurrent events, to define how these intercurrent events exist in relation to the treatment effect in studies. So, as I said, intercurrent events, very important, not easy to understand, but definitely worth the time, because majority of future clinical trials will have a special focus on these intercurrent events and the whole estimand definition that we use. So I think it's very valuable for the audience to focus on this segment. As I said, we will be discussing other segments, but focus on intercurrent events. And we'll also be answering your questions about them. I'm sure that there will be many questions about intercurrent events. How do we deal with them? Do we not impute the missing data, and so on? And it's a very interesting topic. So I would leave it to you, Gareth, if you have anything to. Robert, apologies.
You're fine. You're fine. Yeah, no, that's great. So I think getting some feedback from your mic, Darko. Would you mind going on mute? Thank you. Perfect. Okay. No, I think so. Darko, absolutely an excellent reminder. Please, audience, questions in the chat as we go through this. Excuse me. As we go through this, please start adding your questions. What do you want to hear more about? Because we have a lot of time reserved at the end for answering your questions. So if you've heard of Estimands before, please, but you had further questions on them, ask them in the chat now. That will help us to guide the discussion at the end for the 20 minutes we have reserved. But I think what would make sense to do next is to go through the definition of an estimand and to really highlight what makes up an estimand. What do you need to say, this is my estimand and this is what I'm going to calculate at the end of my clinical trial? So let me share my screen again, because I have a couple of slides here that kind of walk us through exactly what goes into an estimand. So let me do this, 2 seconds. Okay, so what is an estimand made out of? There's really 5 attributes that go into defining an estimand. I have 4 of them, or no, I have all 5 listed here, but we're going to go through the intercurrent events on a different slide because that's really the bulk of it. But I'm getting ahead of myself. So walking very slowly through this. In order to define an estimand, and I should note, you're doing this, you're defining the estimand at study start, at study development, not study start, study development. It's not something that you just calculate at the end of a clinical trial. You hear the word estimand and you probably think, oh, that's just some new statistical output that you create. Not the truth, not the case at all. The estimand is a framework that you develop at the start of your thinking that you want to run a clinical trial. So really the first thing that you have to do is to say, what am I running the clinical trial for? What is my objective? And from there, you then start to list out these 5 attributes of your estimand. So the first one, very simple. You just list out the treatments. What treatment do I want to study? Do I want to go against a placebo? Do I want to go against a reference drug? And what is my treatment? What are the drugs that I'm going to be testing? From there, you then define the population. And this is where things get a little bit more complicated. So the population, yes, it's defined from the inclusion and exclusion criteria. Who do I want in? Who do I want out? But really what you have to do is start thinking a lot closer, a lot more about those results that you're going to want to interpret. Because if you calculate your analysis on the wrong population, you might get a result, you might get that hazard ratio that Darko was talking about earlier, but it's about a different population. You didn't study the correct one that you wanted to in order to answer your ultimate question to achieve your objective. So defining the population is critical. Darko, I saw you come off mute.
I have one very interesting example to complement what you were just saying, Robert. And that is, for example, if a study is about evaluating the treatment effect for oncology area, for example, a phase 2 cancer, the treatment effect, for example, for progression-free survival for phase 2 would be a whole different story than progression-free survival, for example, for phase 3 or phase 4, right? So the biological context is completely different. Progressing from phase 2 to phase 3 is not the same as progressing from phase 3 to phase 4, right? So here's just one example how population is very important. Having a hazard ratio is important, but having the biological context in an estimate is extremely important as well. Sorry, Robert, please go ahead. Just felt the need to add this example, which complements what you were saying.
No, it's a great it's a great addition because the phase of trial absolutely matters. Am I studying on healthy volunteers? Am I studying on a much smaller phase 2 population, and I'm still doing dose discovery, for example, or not dose discovery, dose decisions. So after population is determined, then you move into the variable, and this is the third attribute that defines the estimand. And you really want to be very specific with the variable or the measure that you're going to analyze at the end of the trial to hopefully achieve your objective. So this is, okay, if I'm going to do hazard ratio, what is it that I'm specifically calculating that ratio on? Is it the number of subjects who experience a specific adverse event? The list can go on, but you have to be very specific about the variable that you want to include in your estimand. Fourth on the list is the population level summary. This is your statistic. This is you determining what type of test, what type of analysis do I need to run in order to achieve my objective. And then finally, there is the ICEs. And this is actually a two-step process. So first, you have to list out what type of ICE do you think is likely to occur? Do they have the potential to withdraw from the treatment prior to the final measure, prior to recording the final measure? If that's going to happen, then the second step of this process is, how will I address that? How will I handle that? So defining the ICEs is a two-step process. First is, what can possibly happen to my subjects in my trial? And the second step then is, how do I handle that? And on this next slide that I have for us, these are the policies that are recommended and listed specifically in the ICH E9 Revision 1 guidance. But there are more ways to handle estimands. This is just the ways that are listed out in the specific guidance document that released estimands to the world in 2020 officially. So kind of going in order here, these are they go from very simple to a little bit more complex. Treatment policy, it's basically like the ITT, the intent to treat. It's a little bit different, but really what you're doing here if you follow the treatment policy strategy for handling an ICE, basically what you're saying is, I ignore the ICE. So it's very simple. It's almost like you're disregarding the estimand. You're not. You're certainly laying things out, and it's good to put it forward. But it's the most basic of the methods to handle an ICE. The next is this hypothetical strategy. This is where you would say, okay, the ICE has occurred. The patient has withdrawn from the study, or the patient has taken this new medication that they weren't supposed to. And then in this hypothetical strategy, what you're saying is, hypothetically speaking, let's say that the ICE didn't happen to the subject. What does that subject's data then look like? And so this is where Darko was saying earlier about multiple imputation, or some type of simulation method can be used to say, hypothetically speaking, this is what this subject's data should have looked like had that ICE not occurred. The composite variable strategy, a little bit more complex here, where you are basically saying, okay, the ICE occurred. I'm going to roll that up into the measure somehow, some way. I'm going to say, okay, this occurred. It's going to weight the result, or it's going to change the results that I recorded by X amount. And so composite means to put together. You're basically saying the ICE occurred. I acknowledge that. And I'm going to put it together with the actual observed nature of my data, of what actually happened to that subject. So a lot of policies, and I don't want to go into all of them, but and please feel free to ask questions in the chat if you'd like to learn more about these. But really what we're saying here with the estimands is, the ICE occurs. Tell me how you're going to address that. Tell me how your analysis is going to be changed to address the ICE from occurring, so that you still truly answer the question that you set out to answer first. With that, I'll stop there. Darko, would love to bring you back in. What do you have to add? What would you like to add about estimands and how we handle the ICEs? Oh, Darko, I got you on mute.
Okay. Can you hear me now?
Yes.
Great. Actually, I think that the framework that you just explained is really important, because these are the two most important segments to assess when designing estimates. So those 5 segments listed in the international clinical research documents, such as the one we mentioned before, E9R1, and especially paying focus on intercurrent event strategies that you are just showing. So previously, in past decades, in clinical trials, there were 2 approaches that a lot of researchers were taking. So you mentioned them, the ITT, so intention to treat, and per protocol strategies. So you see, the estimands are built on top of these, the estimand intercurrent event strategies. That's very important for our audience to understand. And some of these actually correspond. So, for example, treatment policy will correspond to ITT principle, and the while on treatment may correspond to other approaches. So, as per protocol, specialized approaches. And the approach is very important because it defines how do we interpret results. So, for example, we need the ITT principle because we don't want to break the randomization in the trial, right? So we want to have an assessment that would correspond more to a real world scenario, because in real world treatments that happen in hospitals, intercurrent events also happen. So they happen in clinical trials, they happen in real world evidence, they happen in real world medical systems. So that's why ITTs are very important, and in this case the treatment policy estimate would correspond to it. On the other hand, sometimes we may want to focus on a scenario where the treatment would be 100% respected in relation to protocols and adherence. Now, this is, as you said, in reality, this is almost impossible, but we may have strategies to try to estimate those scenarios. And one of them is the hypothetical strategy that you were mentioning. Okay, I also have a few slides that I may share for the audience. So let me know once you can see my screen. So it should be available now. Okay. So
can you go to presentation mode?
Okay. So the segments that Robert was explaining are summarized in this image. So you can see biological, statistical segments, and the research questions. So we start with the research questions, and all these segments are interconnected. So the estimand is not just counting or defining different segments in a table, it's actually about integrating them. And this is what Robert also mentioned before. So integrating them into a connected system. And on the right, you may see that estimand is actually an integration of multiple definitions, such as IC, treatment population variable, and population level summaries. So they're connected to each other. And let me just show a few examples. For example, population that I was mentioning before, I may relate to that. So is it the phase 2 cancer?
Darko, have you gone to the, sorry to interrupt, have you gone to the next slide or not yet?
Well, this is the initial slide.
Excellent. Sorry, it's just still not in presentation mode, so I wanted to make sure you hadn't switched slides on us.
Okay, thank you for pointing that out. So the population may affect the end result, the population level summary. So is it a phase 2 cancer? Is it progression-free survival? For example, a variable would be progression-free survival in oncology or overall survival. And then it would be summarized as a population level summary with hazard ratio, for example, in a clinical trial. But the population may change the hazard ratio, right? So different populations will have different hazard ratios. And obviously, when we estimate a hazard ratio, we must relate it to a specific population. And that way, we know exactly what the result is about. So you see, they're related, right? Same goes for ICEs, right? Changing an ICE approach can also change the population level summary. Population level summary is basically the end estimate that we get when working with Estimands. And here's another slide to show you how this looks when it's defined in a clinical trial protocol or even in a manuscript that is published. So this is not the real uh, this is not the real study. This is something that I created as an example of how it would look. So it resembles something that would be similar to a real study. So you can see on the left, we have these 5 segments.
Oh, Darko, sorry. The slide is still the one that you're sharing is still the triangle slide.
Okay, let me just see. Is it changed now?
No, maybe stop sharing your screen and then reshare the window.
Okay, I'll do that.
Yeah.
Just one moment.
And while Darko does that, again, a friendly reminder, please, this is your chance to put questions into the chat. We absolutely want to answer all your questions about Estimands. So please feel free to add your questions to the chat.
Okay, so the new slide should be on.
Yes.
Yes. Okay, so I'm going to repeat a few words because the slide was still left at the old one. So this is how estimand resembling a real world definition in a study may look like. So it might look like a very complicated table, but it's not that complicated. It actually has those five segments that me and Robert are talking about the whole podcast. And then as we go to the right, we have more and more detailed description of what those are. So for example, the intercurrent event strategy, we define it as treatment policy as example. And then we explain what that means. It means that rescue medication will be treated in a specific way. Other intercurrent events will also be treated in a specific way. In this case, it's practically ignoring them and collecting data for the subjects regardless of the intercurrent event happening. And then we may also add additional characteristics. So I added here that it's based on ITT principles, which means that intercurrent events will not mean not collecting data for those subjects. This is very important. Let's just take one angle, because we don't, in some randomized control trials, we don't want to break the randomization. So the allocation of the subjects is the same regardless of the intercurrent events. And this is what intention to treat principle is all about. So it's about those subjects that were originally planned to be treated and followed up, regardless of the non-adherence, the intercurrent events, the rescue medication, and so on. So this would be one example how to define, for example, an intercurrent event strategy. Defining the rest is easier, right? So basically, these are all segments that most researchers are defining in their studies. They're just not always integrating them. So the treatment, what is the compound used, what are the arms, what is the control or standard of care, how do we define these, what is the specific population. This specific example I'm showing here is about cardiology. So you may see the population is, for example, population with specific ejection fraction, just for the audience, the ejection fraction is a measure of how strong is the heart pumping blood during the heartbeats and so on. So then I define this ejection fraction as a variable, as a biological variable. I explain what it is. I explain how to mathematically define it, right? What is it? What is its formula? And so on. And in the end, I define the population summary as mean difference or delta ejection fraction. So I try to go over this estimate very quickly, even though it looks complicated. It's not that complicated. The similar principle would be if we were working in oncology definitions, but the variables, population summaries would be different. As I mentioned before, there would be mostly hazard ratios, progression-free survival, overall survival, and so on. So I'm going to stop here. And Robert, if you want to add, please.
No, I think that was a great overview, and I think it leads nicely into like a real example. So if you don't mind, I'll share my screen, and
Of
we can go
course.
through what we've actually seen in the clinic. So thank you very much for sharing that, Darko. Very helpful. Let me know when you can see this window.
I can see your window.
Excellent, thank you. So very quickly, we had a very nice. I apologize to the person who asked this question, but we had a very nice question come in prior to the LinkedIn Live event going live, and that question was, "Hey, I'm a medical writer. Can you please talk to us about where Estimands live in protocols?" And there is this new ICH M11 template that came out. I forget the date, but basically what I'm trying to show on this screen before I dive into the real example is that Estimands are part of the standard protocol templates that are coming out nowadays. And so it is on the clinical trial design team to include the Estimands for that specific trial in the protocol. And to kind of deviate from that slightly, there are specific therapeutic area specific guidance that now call for Estimands. I don't have any on the slides, but I can give you a list if that's of interest to you of those therapeutic areas that do have specific guidance from the FDA. I'm referencing the FDA because that's the regulatory body that I'm the most familiar with and interact with on the most frequent basis. But they have specific guidances about, okay, you're running an obesity trial, for example. This is the type of Estimand we would like to see. These are the type of treatment policies that we need to see, or ICE policies, excuse me, that we need to see included in your protocols. So just a quick note before I dive into the example here, but there are Estimand sections in protocols. Okay, the example that I have comes to us from Pulverijan, and I apologize if I butchered that name, but they released an article a couple years ago about a practical guide for study teams with examples based on psychiatric disorders. So they're giving us Estimand examples for psychiatric disorders, and the one that I've chosen to present today is one around major depressive disorder. So Estimands always start with the question. They always start with, what is the question you're trying to answer? So the question we very specifically want to answer in this example is, for a patient with MDD, major depressive disorder, for whom acute drug monotherapy would be indicated, what would be the expected effect if I gave them drug X, I've removed the drug name, on a depression severity scale at week 8, where no antidepressant medications are available? So the key takeaway here from this question is to note how specific it is. It's no longer okay to just say, hey, my question is, does my drug work on patients X, Y, and Z? You now must say very specifically, who is it for? What is the specific question you're trying to ask and answer? And so being very specific in your questions is critical to setting up stronger Estimands. And so in order to answer this question, we are going to conduct, in this make-believe example, in this real-world example, we are going to conduct a parallel, double-blind, placebo-controlled randomized trial, and we are going to use Estimands for the primary objective. Okay, so what we're going to do next is go through the specific attributes to create the Estimand. So this is us pretending that we are going to create this Estimand, which was already created, and this is how we would do it. So first we would list the treatments. For this one, I've removed them because it's drug X versus placebo. And next we go in and we define the population. So in this one, we've already answered the population question by having our question, the question we want to answer, being very specific, right? We know that it's patients who have been diagnosed with major depressive disorder. And even further than that, we also want them to be currently experiencing a major depressive episode with at least moderate symptom severity. So we're setting our baseline for where we want to begin our assessment. If somebody has too low of symptom severity, it's probably going to be difficult to detect the difference that we're looking for at week 8. So we're being very specific with those types of patients that we will enroll in this trial. Next, we move down to the variable, and we're going to simply define it as the change from baseline at week 8 in the total score of the HDRS, the Hamilton Depression Rating Score, which is a 17-item rating system that will be able to determine what is that subject's change from baseline at week 8 on. And then what type of statistical analysis are we going to use? For this one, we're going to use a difference in means between the two treatment conditions. And we'll talk about the statistical models that we're going to run in a little bit. But now the main attribute that we have to discuss is the ICEs. And again, this is a 2-step process. So this first slide is only covering the first step. And that first step is identifying what ICEs are likely to occur. And for this example, there's been 2 that have been identified. Either the subject can discontinue from therapy, or they can start some other treatment for MDD. If either of those things or both of them happen, it's going to muddle the question that we've asked. Because now if that happens, if the subject discontinues from the treatment, the answer to our question of what does my drug do at week 8 can no longer be answered accurately because the subject discontinued from treatment. I don't know. Or if they start different therapy, then the question I'm answering becomes, what does drug X plus this new treatment do to the MDD score at week 8? So we've identified 2 possible ICEs. Now we want to determine how are we going to handle them. And that looks like this. So I also use a table just like Darko did. So here on the leftmost column, I have the ICEs that we've identified. Again, that's treatment discontinuation or starting a second drug. And then I start to define the strategies. So if you go back to the slide that I had where we were listing out the potential solutions that ICH E9 provides, they mentioned treatment policy and hypothetical. So those are the 2 we're going to look at for this example. But what do they really mean? When I say that we are going to handle the therapy discontinuation ICE by using the treatment policy, really what we're saying is disregard the ICE. I pretend that it didn't happen. I will still take the data as it is and analyze it as it is. And maybe that's okay. Maybe we're saying that because we want to be harsh in our assessment of the drug. It's doing this is if the subjects discontinue from our drug, from the drug that we're studying first before the analysis, it's obviously going to be a negative impact on that week 8 assessment. And then the second second strategy, that we're going to use to account for if the subject starts a different pharmacological treatment is to say the hypothetical strategy, which again, if you recall, is, okay, the ICE has happened, but I'm going to imagine a scenario where the ICE didn't happen. And so the key elements here of these 2 different strategies is that you're still collecting efficacy data the whole way through, but in the therapy discontinuation ICE, I'm still going to use the data that I collected on that subject after they discontinued from the treatment, if I was able to still collect that data. Whereas the hypothetical strategy, I'm not going to use it. So I'm not going to use that data. So if the subject starts a new treatment in addition to drug X that we're studying, I'm now going to say hypothetically that they didn't start that treatment, which means I'm no longer using the data that we collected on that subject after they started their treatment of this new treatment specifically for the assessment of my primary endpoint. So really what this looks like, and this is what Darko and I were discussing prior to this call, is different uses of imputation of data. So in the therapy discontinuation ICE, I'm not necessarily going to impute any data for my Estimand calculation. I might still impute data if there's enough missing data at random, but I'm not going to be imputing any data because of the ICE occurring. Versus the if the subject starts the other treatment in addition to drug X, I am going to impute data and I am going to use that impute data. So a lot of nuances here, and I know I'm going very quickly, but I do want to leave some time for questions at the end. So just to kind of wrap this up, really what this means is I'm using the Estimand framework to specify to the reviewers of my clinical trial that I handled the data that I collected slightly differently based on which ICE occurred. And I'm really detailing that out in the protocol, and I'm really detailing that out in the Estimand section of the study protocol, so that when I report the results, when I actually say, hey, this is what happened and this is the actual statistic, in this case, we would have used an ANCOVA for these analyses, I can say I report that statistic. So when you report an Estimand, you're still just reporting the statistics you calculate, but you're adding the additional information of what was the population, what were the ICEs I knew were going to occur or predicted would occur, and how did I handle those ICEs? How did I handle that data? So I know I went very quickly. I want to leave some time, A, for Darko to address this example and to add anything that I might have missed. And then B, let's dive into the questions. So Darko, please, if you could come off mute and let us know your thoughts about this example, that'd be great.
Well, I'll just add a few real world examples about a very important topic that we just discussed earlier yesterday and today, which is the missing data imputation. So we may need to impute the data in many scenarios. So in multiple scenarios on multiple ICA strategies, we may need to impute data, but for different reasons. So as Robert explained, in the treatment policy scenario, we don't impute the data because of the ICEs, right? If we impute data, we would impute it for a different reason, such as a technical reason, right? Inability to collect data for some reason. It could be a data management system. It could be some unpredictable event outside the clinical trial. It could be patients changing locations, and for that reason, their data cannot be collected. So the reason for imputing data is very important. And on the other hand, in a hypothetical strategy and some other strategies, we may impute data because it's actually inherently defined in the estimate. So the hypothetical strategy actually means that we have to impute the data to get to that hypothetical scenario. I mean, as always, there are cases where this doesn't have to be done, idealistic cases, but most of the time we have to impute the data to get to this hypothetical scenario. And since we are at the last phase of the podcast, we have some very interesting questions which are actually related to this. So we may answer them. So there's a question about deciding the strategies related to ICEs from Guppy. So how do we decide the strategies? And me and Robert mentioned some of these during the podcast, some of these aspects, but I will now summarize them. So obviously we start with a research question. We start with the main goal of the study. What are we studying? So for example, if we want to study the causal treatment in a randomized phase 3 controlled trial, and we want to preserve the randomization, we want to use the bias correcting effects of the randomization, which are very important, then we would use a treatment policy estimate, for example. We can use other estimates as well, but treatment policy is frequently used in this situation. And if we were to, for example, be very accurate about pharmacodynamics, for example, we may want to use hypothetical estimate because we are interested to see exactly, accurately, how would the pharmacodynamics behave in case a certain number of subjects or patients were to take a specific drug or be on a specific treatment. So I hope that helps answering your question, and I'm gonna leave it to Robert
Yeah.
to uh, compliment.
Thanks, Darko. Again, some reason getting feedback from your mic. Would you mind going on mute?
Okay.
Thank you. Thank you. It's a great question. It is the question when it comes to determining your estimand and how you're going to frame it. So Darko's response is spot on. You start with the end in mind. Everything about the estimand comes down to the question you want to answer and how close you can get to answering that question specifically. So statistics is this really crazy thing that is the bridge between the real world and mathematics, right? And so we as statisticians are positioned very nicely to set up the estimand in such a way that we can say, okay, if these ICEs are likely to occur, we can use these strategies to handle what that means, what the ICE occurring means for the data in such a way that we can then begin to answer the question more accurately. But the key thing here, and we haven't touched on this yet, and I apologize, the key thing here is that estimands are not built just by statisticians. Absolutely, the statistician is one of the main contributors, but so is the clinician, and so is your regulatory team. So you need to, as the statistician in this example, you need to work very closely with your clinician to say, hey, what ICEs do you think are likely to occur to these patients? We have them going through this protocol. What's likely going to happen to them? And then from there, you as the statistician can determine, okay, based off of what these things are going to happen and what that means for the data, I need to address the data in X, Y, and Z way. I can ignore the data. I can impute new data based off of prior beliefs. I can do a composite variable to make it a more meaningful, direct answer to the question, whatever it might be. You, as a statistician, have that ability to come up with the solution, the handling solution, the policy for the ICEs that are likely to occur. So I hope that answers your question. I know it's a very vague answer, but it truly is dependent upon the question that you want to answer and what you believe is going to happen to your data, and then how you need to address or how you need to help that data to be corrected in such a way that it answers the question a little bit more directly. Okay, so we had a question earlier from Alex about, can you elaborate on how estimands relate to what's traditionally been done and what is new? It seems like these things are what we should have been considering all along, but maybe it's just a formalization of it. Alex, spot on, 100%. This is absolutely something that we should have been doing this entire time, and the estimand framework is really the ICH's way of saying you got to do it better. You got to be specific with it. You have to really say, what is the question you're asking? Who are you asking it of? What's going to happen to your subjects? And how are you going to address those things that happen to your subjects? So from my standpoint, really the only things that are quote unquote new when it comes to estimands is the way that you are consolidating that information and putting it into a very readable and systematic way so that it's reproducible across all studies. So really what this is helping to do is to really foster meta-analyses in a better way in the future, because how easy would it be for me to say, oh, I'll look at this study, and this study, and they all have their estimands laid out. Now I have a very specific way of saying, yes, these studies are asking and answering the question that I'm concerned about with my meta-analysis. Versus if you're doing that for studies that don't have estimands, you have to do a deep dive to ask yourself, how did they address missing data? How did they address the ICEs that occurred? All that stuff. So long-winded way of saying, I agree with you, Alex. This is a much more structured way of saying, I've thought about these things and here's how I'm going to account for them. Darko, anything to add? Do you agree with my statement? Oh, Darko, still got you on mute.
Okay, I absolutely agree. I think it's really a fantastic statement, and I would add a specific answer, which is of course related to this, but it has another angle, which is are estimands 100% required in terms of the legal and regulatory perspective? Well, I will answer it like this, and Robert can, of course, add his view. So, while there are clinical trials, they have been, of course, in the past, and other studies which don't specifically have estimates. Estimates are very important and beneficial because they are considered a part of the good clinical practice. So, they are defined by the regulatory agencies such as FDA and EMA, as a part of good clinical and good statistical practice in their documents. So obviously, having estimate specifically defined in your clinical trial protocol design, implementation, and so on is definitely beneficial and important for your study. Now, whether it's going to be mandatory, as asked in the question, well, it's probably up to different regulatory agencies around the world. There may be differences, but I believe that it will be part of the good clinical practice in majority of the continents and in majority of jurisdictions for the regulatory agencies. Do you have anything to add, Robert?
I think that was perfect, Darko. Totally agree. Brilliant. Okay, I saw one more question from Gopi come in, and thank you for asking the questions, Gopi. Phenomenal. And he's asking, do we have any list of ICEs specific in each therapeutic area defined by regulators like the FDA? Darko, I am not, and I believe the reason why is because ICEs are specific to that protocol. So you have to think about what's going to happen to your patient on their journey through your protocol. And so even though I'm running a protocol, for example, on obesity, maybe it's a different protocol. It's a different way of doing that. Maybe it's a crossover study. And so now I've introduced the ability for a subject to fail to cross over versus a parallel design. So you really have to think about what's going to happen to your patient in order to determine what ICEs are available. What regulators do provide and what the International Council for Harmonization has provided is the strategies to handle those ICEs. And that's what we were showing on the earlier slides about there's the treatment policy, there's the hypothetical strategy. So those can be standardized and a list can be created. And I think we're adding to that list as we move forward with the Estimand framework. But long-winded way of saying, no, not to the best of my knowledge, there are no specific lists of ICEs. Darko, are you aware of any?
Well, I think you answered the question correctly, Robert. So it's dependent on every area, and there are several most used strategies, and these strategies can be used in multiple therapeutic areas. So again, it depends on the trial and it depends on other aspects. Maybe one additional angle that I can add to this fantastic explanation by Robert is that even though there are no specific strategies in terms of one therapeutic area for one specific strategy, segments of the estimates can be relevant for some therapeutic areas. So, for example, a population level summary. In oncology, we may use hazard ratios most of the time, or we may use other population summaries in other therapeutic areas. So this was just one example. But I think that Robert's point is correct, and that it's dependent on every therapeutic area. And ICEs have multiple therapeutic areas where they can be used. So they're not strictly bound to specific therapeutic areas, but more to specific research questions.
Brilliant. Brilliant. Okay. I think we've answered all the questions. Darko, do you see any others?
I think we answered the questions that we could address live. Obviously, if there are any other questions that we didn't answer in this moment, we will answer them after the podcast. So we will go through each question and answer them. So any comment or question that you ask will be addressed, and we will try to do it in as short period of time as possible.
Excellent. Darko, I want to say thank you very much for the time today. Excellent discussion on a very critical topic that only is going to gain in importance over time. So thank you for your time today. I hope everybody on the call was able to take something away. But thank you all for joining us. Darko, any parting words?
Thank you so much, Robert. It was really great to discuss all these important aspects with you. Thank you for your time, and looking forward to discussing this even further. And we will, of course, discuss with the audience as well. So we'll be communicating with everyone who interacted with the podcast.
Brilliant. Thank you all. Have a great day.
Thank you all.
Till next time.
Have a great day.